Perfect. So forensic genetics stands at the crossroad of science and law, because forensic genetics use the language of genetics to resolve mysteries and help to find the truth in the legal investigation. And this kind of discipline is based on the gold standard that today is represented by the analysis of DNA, and permit, in this context, to identify specific individuals in a univoc way and distinguish each individual from another. In other terms, forensic genetics aim to identify with the greatest possible precision the biological origin of a sample to help the justice system and solve both criminal and civil cases. The application of forensic genetics is not only the simple identification, because it extended the resolution of biological relationships, and through parentage testing and kinship analysis, geneticists unravel the intricate familial ties, aiding in resolving paternity disputes, locating missing persons, and establishing family connection crucial in legal proceedings. Forensic genetics realized the marriage between science and justice, revolutionary.
Sorry, Prof.
Yes.
The slide are not moved.
Ah, sorry. What's happened? I try again to
share. Yes.
To share. Sorry for the inconvenience. I start with this. Okay. Let me say.
Try.
No, it doesn't move. Okay, now in this way.
Okay. Yes. Yes.
Okay. Okay.
Perfect.
Okay. I have to use the. Sorry, I have to use the mouse. Forensic genetics, as I say. Okay, and it has an important role in identifying perpetrators, exonerating the innocent, and renewing families. Underscore its pivotal role in the quest for truth and fairness within the legal system. Nonetheless, as the field advances, it is important to avoid to forget the ethical and legal complexities, ensuring that justice is served while respect of individual privacy and rights. So, at the first glance, the ethical and legal implication of using genetic information in forensics might be underestimated, but the collection, storage, and use of DNA data raise concern about privacy, consent, potential misuse, and striking a balance between the pursuit of justice and safeguarding individual rights remain an ongoing challenge in this field, requiring continual refinement of legal framework and ethical guidelines. A continuous evolution characterized the history of forensic genetics, not only in the type of biological marker, but also from the point of view of techniques and methodology used. So the goal is to increase application and discrimination power efficacy. DNA deposit sources are not limited to the obvious biological material as blood, tissue, stains, fluid, but also include other kind of items that may have received body fluid or skin cells. Personal item, for example, handled regularly, often contain skin cell deposits that are an excellent DNA source. Item touched or handled repeatedly, or it is in close daily contact with an individual, are potentially rich sources for DNA. And items also discarded by a suspect at crime scenes, such as drink containers, gum, hats, masks, and bandanas, should be documented, collected, packaged, and sent to the laboratory for DNA analysis. DNA is the desoxyribonucleic acid and is a major component of the chromosome found in the nucleus of each cell in the body and also in the human body. And are the steps in which we works to obtain at the end the results of the DNA analysis. It is performed in paternity testing, forensic casework, DNA databasing, missing person testing, lineage studies, identification of remains, and so on. And what we have at the beginning is the sample that are collected in the crime scene during the crime scene investigation. Then we have in the lab the DNA extraction, the quantification, the amplification of DNA, the separation by electrophoresis, and at the end, the interpretation. So I don't want to enter in the technical aspects, but we have to highlight some assumption about the genetic profiles. The genetic profile, the result of the analysis of DNA. So each individual has unique DNA profile, and each person is the same DNA profile in every cell of the body. An individual's profile remain the same through the entire life, and half of your DNA comes from your mother, and half from your DNA comes from the father. So regarding the forensic genetic application, we have a different application in the routine cases. The first one, and is the I can say the more famous, is the crime scene analysis, famous for the series TV in the that we can see at the television. And crime scene analysis is when the forensic genetics is often used to analyze biological sample collected from crime scenes with the purpose to compare the genetic profile with a suspect or a victim involved in this crime or eliminate the individual for consideration. Another aspect that is similar but have a different purposes is the identification of human remains that plays a crucial role in identifying human remains, especially in cases of mass disaster, accidents, or unidentified bodies. And this is essential for providing closure to families and for legal and humanitarian reasons. And then paternity testing, in which forensic genetics is commonly involved in paternity testing to determine biological relationships between individuals. And this is a legal implication in case of related to child custody, inheritance, and child support. Coming back to the crime scene analysis, and this is an overview of what I very simple explained in the past slides. We have the extraction, the amplification, the analysis through this instrument that is a particular instrument used, and this is the result. So this is what we can call genetic profile. And there is a principle that is called the Lockhart principle, that is whenever it goes, the human body leaves traces behind: hair, saliva, other fluids, footprints, touch DNA. And body's materiality has been a key part of criminal investigation through the history. The central law of the human body as the basis to identify authors of crime has gained different shapes in the last 35 years as forensic science became more commonplace in criminal justice systems. Applying forensic genetics involves collecting and analyzing biological evidence to establish connection between individuals and the crime. Biological materials collected from the crime scene are crucial as they contain DNA that can be used for identification. Then DNA is extracted and analyzed to obtain a DNA profile that is a unique genetic profile for each person. If a suspect is identified, DNA can be collected for comparison, and if there's match between the suspect DNA profile and the crime scene sample, it can be a strong piece of evidence in linking the individual to the crime. Differently, when the suspect is not identified, the DNA profile obtained from crime scenes are compared against local, national, and international DNA databases. If a match is found, it could link DNA to an individual already contained in the database or provide investigation leads by identifying potential suspects or previous offenders. Forensic genetics analysis is crucial to various criminal cases where biological evidence is present. Here are the following slide. I will present some scenarios relating to the forensic science where DNA play an important role. Sexual assault cases, DNA analysis helps identify perpetrators by matching their DNA with samples collected from the crime scene or the victim's body or belongings. Homicides and violent crimes, DNA evidence from blood, hair, skin, cell, or other body fluids found at crime scenes can link suspects to the crime or exclude an innocent individual. Property crimes, DNA left behind on items like burglary, tools, mags, or gloves can link suspects to the crime scene. Cold cases, DNA technology advancement allow re-examination of all the evidence, potentially leading to breakthroughs in cases that were previously unsolvable. And last but not least, mass disaster or accidents in events like natural disaster, plane or other vehicle crashes, or mass casualty incidents. DNA analysis aids in identifying victims when traditional methods aren't feasible. In conclusion, relatively to the forensic genetics in crime scene analysis, it plays a pivotal role in identifying suspects, excluding innocent individuals, corroborating witness statements, and reconstructing events. Continuous advancement in technology and methodologies enhance the accuracy and reliability of DNA analysis, contributing to the resolution of criminal investigation. Another aspect that I have shown is the identification of human remains that has the following step in the identification. The sample was collected from human remains. Obviously that we need documentation, a storage of found remains, the examination in the labs, and the comparison with collected ante-mortem information or data from the presumed or possible family. In human remains, what is the useful of DNA in this context? For example, in cases involving cases with the corpse are highly fragmented, and DNA provides an essential component of the identification process because DNA can permit to identify the victims, associate fragments and remains, and assist on ongoing medical and legal investigation. Around this aspect, there is a lot of ethical and respect of human rights, and for the situation of time, I have no time enough to explain this aspect, but that is an important aspect of the usefulness of DNA in this context. And last but not least, we have paternity testing. Paternity testing is a fundamental application of forensic genetics, involves determining the biological relationship between a child and a father, alleged father. The process relies on analyzing DNA sample to assess genetic similarities between individuals. Paternity testing has significant implication in legal matters related to child custody, support, inheritance, and establishing familial relationships. The accuracy and the reliability of DNA analysis have made a trusted method for resolving paternity disputes and confirming biological parentage. For this reason, some factors to consider regard accuracy, legal versus non-legal tests, consent to examination, testing on relative are really important. So paternity test results can have a significant emotional implication for those involved. It is important to handle the interpretation of results with care and sensitivity, especially when discussing them to those affected. And this is the important role of the examiner and the relationship with the person that asks for this kind of examination. Also, in this case, we have deontological and ethical duty in relation to this person or patient, like you prefer to call them. And now we move to the final part of my brief talk regarding the future of forensic genetics. In the last 10 years, forensic genetics focus on developing additional traits for unidentified specimens to reduce the number of suspects, generate fresh leads in cold cases, or identify an unknown person or mass casualty victims. And these methods are completely different now from what we are examining and research until this period, current years. So we have moved to other kind of markers that are markers related to visible characteristics like the color of the eye, the color of the skin, the hair. And also we are trying to develop research and obviously to apply this kind of research into the routine using market related to the biogeographic heritage. So from which part of the world the person that leave the stains comes. And this group of markers that are called forensic DNA phenotyping use genetics associated with certain physical traits to generate estimation of an individual's appearance. And this term is associated with the predicting or determining certain visible physical trait, as I said, but also facial features from DNA evidence without relying on direct observation. In essence, FDP outcomes can serve as biological witness and may potentially provide even more accurate information than human aid witness do, who are known to be unreliable. As such, FDP is expected to provide investigative leads allowing to trace unknown perpetrators who are not identifiable by conventional comparative DNA profiling. And PDP is also expected to be useful for missing persons identification in case where reference DNA profile from putative antemortem sample or from putative relatives are unavailable. And this are the scenario that I presented regarding a brief, very brief technical aspect regarding forensic genetics. And I have to say many thanks for your attention. And now I open the floor to the eventually questions, curiosities, comments, whatever you think that could be useful for the person that attended to the meeting, and also for me, for give the opportunity to answer to your question. Many thanks for your attention.
Thank you very much, Professor, for your interesting and very clear presentation. If anyone have a question, you can write this in the section for question and answer that you found in the Zoom. I'm looking if there is already some question. Not for the moment. In the meantime that we're waiting for your question, if we have, I have one question that is not directly related to the presentation, but that I believe that could be helpful for our listening. And you, as an expert in your field, having seen the program of our master, how can this master? How can you think about this master? And how this can adding a value for the student to perform your activity?
Okay. Forensic genetics is one of the forensic sciences in the landscape of forensic medicine. What is the point? That we have some courses. My experience is Italian experience, and we have some degree courses that is called forensic biology or something like this. What I think that would be the adding value of this master in which that I have the opportunity to see the program is the fact that is particularly focused and also to give some technical information to person that undoubtedly with a degree in biology can have the expertise to extract DNA, to amplify DNA, but in this case, they are focused on the use of DNA for a forensic purpose. And as I said in the beginning of my talk, we have different biological stains, we have different biological material, so we have to be able to adapt the technique that are using worldwide to this particular aspect of genetics, first one. And this is the technical approach that probably will be more focused in a master of this kind in relation to a degree course in general forensic. And the second aspect is the one researcher or a scientist or a doctor like me works in this aspect have to take in consideration also ethical and human rights aspects related in how to conduct the analysis, how to present the analysis to the court, and how to perform the analysis in a correct way, just to respect the interest of justice and the human right of the person. And this is all aspects that are strongly taken in consideration in this master. So I think that this is the goal of this master in this aspect of genetics. So we have a question?
Yes, we have a question.
I
How
have a question.
accurate is phenotypic DNA profiling, and what are some ethical concerns regarding the future development in forensic genetics?
Okay. At the moment, FDP or AVC is depend also is two different ways to call the same things. Have some in some European states they are permitted. In other European states, there is not a permission to use them because at the moment they are not much accurate. In the meaning is that we have not to study not only one gene involved in this, but more than one gene, and this is difficulties. At the moment, they works very well with the extreme condition: black skin, black hair, black eyes, blonde hair, blue eyes, pale skin. In the middle, we have difficulties to attribute in a perfect situation the characteristics and to with a, I can say, with a correct position to go to discuss in the court. So we are not so precious, perfect and accurate in the center characteristic that stay in the middle of the two extreme situation. Regarding the ethical concern is related to the fact that when I do not have a genetic profile of the stains, but I have to identify some visible characteristics. So for example, I say these stains come from a blonde woman with blue eyes and pale skin. So the first thing is that this person can change this evidence and these evident characteristics. So I'm looking for one person, but I am not able to find this because in this case, this woman changed. But above all, there is the fact that I do not identify a precise, a unique individual, but a group of individuals. And these individuals have to escape from this indication, have to defend themselves to this situation. And this is not perfect correctly to work in this way from a point of view of human rights and from an ethical point. And this is one of these situations in which these characteristics work. I found a group of individuals, not just only one individual. And this is probably in this group, there is sure persons that are not involved in the crime. This also can create inside of a population form of discrimination. And this is another question that have to be deeply investigated in the routinary application of this kind of genetic characteristics in forensic science.
Perfect. I think that the answer was very clear. So we have another question, sorry. What is the shorter time range in which we can give accurate results regarding our specimen? So the short time.
The shorter time
Yes.
is if I start now and I consider starting from DNA for extraction to amplification and analysis with the new technologies that are just a little improving this couple of years. I think that we can 1 hour, 4 hours, no? 4 hours are enough to produce a genetic profile. Okay.
Yes. There are other questions? Someone? No? I think that was all clear. Yes. So, in concluding the master section of forensic genetics, we emphasize the integral link between the discussed topics and the forensic biology master program. Indeed, the above is just a small taste of what will be proposed in the master. This program, offered by the European Forensic Institute, provides students with a deep dive into the forensic biology world, from quality assessment and of biological evidence to DNA analysis and much more. All the modules are essential for understanding the intricate of forensic biology and forensic genetics. The curriculum is meticulous designing to arm students with both theoretical knowledge, but also and mainly practical skill crucial for forensic science careers. through innovative technique methodology and practical exercises, the graduate emerge well equipped to confront real-world challenges in DNA analysis, crime machine investigation, and navigate in the ethical dilemmas with the legal framework, as the professor said before. And the Master of Science in Forensic Biology not only deliver a comprehensive education, but also foster an environmental conducive to ongoing learning and adapting within the dynamic field of forensic genetics. Once again, thank you to Professor Caenazzo and to all of you. For any question regarding the program of the master, I would like to inform you that you can book a short individual meeting on the website of the European Forensic Institute to further discuss with me about this master. And moreover, always on the website or on our social media, you can find more information about our upcoming meeting with the other expert. And that's all. Have a good evening to everyone, and thank you again to all for your attention.